In this chapter, we'll be examining the brain and the cranial nerves. We'll begin by looking at an overview of the brain and its main structures, and then the membranes that cover it, the ventricles or chambers within it, the cerebrospinal fluid that bathes the brain, as well as its blood supply. We'll look at the hindbrain, midbrain, forebrain. We'll examine some integrative functions, and then we'll finish off the chapter by looking at the cranial nerves. So, we've looked at the brain for a long time. As far back as Aristotle's time, he thought the brain was more of a radiator and was involved in cooling of the blood. Hippocrates had a little bit more accurate of a view. His statement, "From the brain only arise our pleasures, joys, laughter, jests, as well as our sorrows, pains, griefs, and tears." So even way back in this time, there was some understanding of how the brains function. The brain is so integral to life that it's associated with what it means to be alive and human. So the cessation of brain activity is taken as the clinical criterion of death, even when the other organs of the body continue to function. Now, the evolution of the central nervous system shows the spinal cord has changed very little, while the brain has changed a whole ton. The greatest growth are in the areas of vision, memory, motor control, and then, of course, the prehensile hand, or in the opposable thumb. The brain is simply an extension of the spinal cord. As the spinal cord has a hollow canal down the center, so does the brain. It's basically a highly convoluted tube. We'll explore its structures in more detail coming up here shortly. Let's look at some general directional terms and landmarks on the brain to begin with. Rostral means towards the forehead, and caudal means towards the spinal cord. The brain itself weighs about 1,600 grams, or about 3.5 pounds in men, and about 1,450 grams in women. There are three major portions of the brain. The cerebrum occupies 83% of the space. It's covered with gyri and sulci, which are these bumps and grooves all over the surface, as well as fissures. It may seem at first glance that these bumps and grooves are random. However, they're very consistent from one brain to the next, so much so, in fact, that we can name them. The longitudinal fissure is the deepest gyrus between the two cerebral hemispheres. The cerebellum, although it's fairly small, contains 50% of the neurons in the brain, and it's the second largest region. It sits right in the posterior cranial fossa of the skull. And finally, there's the brainstem region. The brainstem is the portion of the brain that remains if the cerebrum and cerebellum are removed. It contains the diencephalon, the midbrain, the pons, and the medulla oblongata. All of the older functions of the brain are here. That is, all of our involuntary functions that maintain our life. The cerebrum covers our higher levels of processing, and the cerebellum is mostly about coordinating movements. Again, here's a top view of the cerebrum. We can see the longitudinal fissure here, and then these gyri, which are the thick bumps, and sulci, which are the shallow grooves. The corpus callosum is a thick nerve bundle that transfers information from one hemisphere to the other across the bottom of the longitudinal fissure. The cerebellum occupies the posterior cranial fossa. It also has gyri and sulci and fissures. However, they're much smaller than the ones we see in the cerebrum. It occupies about 10% of the brain volume, but contains over 50% of the neurons. Lots of processing occurs in the cerebellum. In this figure, we can see a cross-section of the brain. We see one cerebral hemisphere and this big band of white tissue here. These are myelinated neurons that connect one hemisphere to the other hemisphere. It's called the corpus callosum. Now, as you can see, when we look at the cross-section of the brain here, there are both gray and white areas. The gray matter is where we see all the neuron cell bodies, their dendrites, and their synapses. It has a dull white color when it's fresh due to very little myelin content. It forms the surface layer of the cortex over the cerebrum as well as the cerebellum and forms deep nuclei within the brainstem. The white matter consists of bundles of axons, as exhibited here in the corpus callosum. This is just simply a bundle of axons that moves from one hemisphere over to the other hemisphere. It lies deep to the cortical gray matter and has an opposite relationship in the spinal cord. If you remember, we had gray matter in the middle and white matter around the outside. In the brain, it seems somewhat inside out. The white matter has a pearly white color. This is due to the myelin that's found around each of the nerve fibers. It's composed of tracts, which are bundles of axons that connect one part of the brain to the other, as well as to the spinal cord. You may recall that the nervous system develops from ectoderm, which is the outermost tissue layer of the embryo. Early in the 3rd week of development, we see a dorsal streak of neuroectoderm along the length of the embryo. This thickens to form the neural plate, which you see here. The neural plate is destined to give rise to most of the neurons and all of the glial cells, except for the microglia. They come from mesoderm material. As the thickening of the neural plate progresses, it sinks and its edges thicken and it forms a neural groove. The neural groove has a raised neural fold on either side. The neural fold fuses along the midline, beginning in the cervical region, and progresses rostrally and caudally. By the 4th week of development, this creates a hollow channel called the neural tube. The neural tube separates from the overlying ectoderm and sinks deeper into the tissue. It grows lateral processes that will later form the motor nerve fibers. And the lumen of the neural tube becomes a fluid-filled space, which is the central canal in the spinal cord, as well as the ventricles in the brain. So this is the part of development where we can see both the brain and spinal cord developing as a hollow tube. As development progresses, this hollow tube will fold back on itself in the rostral region to form the brain. Now, neural crest are these cells on the outside in yellow here. This is formed from ectodermal cells that lay along the margins of the groove. They separate from the rest of the tube and form a longitudinal column along each side of it. These will give rise to the 2 inner meninges or membranes that cover the brain and most of the peripheral nervous system, as well as some other structures like the skeletal, integumentary, and endocrine systems. Now, as I had mentioned, this tube is going to start exhibiting some changes. There are 3 anterior dilations that form. These will be the primary vesicles. At this point, they're called the forebrain, the midbrain, and the hindbrain. You can see this tube here. By the 4th week, we see the forebrain, midbrain, and hindbrain. And by the 5th week, it will have divided into 5 secondary vesicles. The forebrain will divide into 2 of them, forming the telencephalon and diencephalon. The telencephalon becomes the cerebral hemispheres, and the diencephalon has optic vesicles that will become the retina of the eye. We can see these 2 regions here. The midbrain will remain undivided and will be known as the mesencephalon. However, the hindbrain will also divide into 2 vesicles, the metencephalon and the myelencephalon. These will be regions of the brainstem that we'll be discussing their functions here shortly. So again, you can see this hollow tube around 4 weeks that gets some specializations in the rostral end. As you can see, the prosencephalon is folding under, and then as the embryo develops, the telencephalon, or the yellow region, which will be the cerebral hemispheres, will grow back over the rest of the developing brain. Take a moment here to pull out your pen and paper and note down all the different regions of the brain. Leave out their functions for now, just the 5 different regions of the brain in order.